All right, so I was just right away. Maybe I'm pretty sure that other people will not be joining because this is exactly what I did in the last semester. If it's the online and if there's no attendance code, I don't bother joining because it's also recorded, right? So I totally understand that. So anyway, I just started right away, and I think I will only be able to finish this chapter for today. And also, I would like to explain detail for chapter 2. Chapter 2 is very easy, and there are not much that you don't need to. There are not much that you need to know, but the water potential is what is hard to understand. And also, as far as I remember, there are questions about water potential. So I want to explain it in detail, and I would like to do that in person. So maybe I will just start for today with these inhibitors, allosteric inhibitors, and so on. So right now I start here. We have a cofactors. So what is the significance of cofactors? Some of the enzymes, they cannot function alone. They cannot walk alone. So what they need is the cofactor. Only when the cofactor bind to this enzyme will be able to change the substrate into the products. So without the cofactors, they will not be able to work together with the substrate. So we need these 3 types of cofactors. And what you need to know is this one. Prostatic root, it will be attached permanently to the enzyme. So that is what you need to know. So it will not be separated from the enzyme. It will be attached to the enzyme forever. For inorganic ion and coenzyme, they are loose. They don't attach to the enzyme permanently, so it's reversible. That's what you need to know. So in other words, cofactors, they can either be attached to the enzyme permanently or loosely. For inorganic ion and coenzyme, they are loosely attached to the enzyme, and prostatic root, they are permanently attached to the enzyme. That is all you need to know. That is it. It's also good to know the azimuth. Inorganic ion, very simple. It's the ion, right? These ions. And then where you can know this azimuth, and also prostatic root is a non-protein molecule, and the heme. In the next semester, if you are going to do the human biology, you will learn more about heme. So heme is attached to the hemoglobin, and it is permanent. It's permanently attached to the here. What is the catalyst? The enzyme catalyst. Heme is present in the enzyme catalyst. Heme is also a pigment that is attached to the hemoglobin. But here, heme is present in the enzyme catalyst, and the enzyme catalyst catalyze the hydrogen peroxide into oxygen and water. Good to know that, all right? If you want to get more marks, of course, you need to have more knowledge so that whenever the questions come, you will be able to answer. For the coenzyme, they are organic molecules, small organic molecules, and they are loosely and temporarily. Like I said, coenzyme and inorganic ion, they are loosely attached and they are reversible. They are not permanently attached. All right, so that's what you need to know for the cofactors. And now we will go to the enzyme inhibition. Enzyme inhibition is something that has to do with the enzyme inhibitors. And those enzyme inhibitors, what do? They will inhibit the activity of the enzyme or slow down. They will either slow down or they will stop the activity. Okay, so how do they do that? We have 2 types of enzyme inhibition, the competitive inhibitors and the non-competitive inhibitors. All right, tell me what is the difference between the competitive inhibitors and non-competitive inhibitors? Competitive and non-inhibited?
Non-competitive inhibitors permanently reduce the max rate of reaction.
That's true. And what about the competitive inhibitor?
No, it'll just take longer for it to reach the maximum rate of reaction.
That's also true. For competitive inhibitor, you can also say the shape of the inhibitor. Let's start with the shape of the inhibitor. So for competitive inhibitor, the shape of it is quite similar to the substrate. So enzyme confuse these two, competitive inhibitor and real substrate. So what the competitive inhibitor does is they compete with the substrate for the active site. We have the active site, right, on the enzyme. And this active site is only for the substrate. But here, competitive inhibitor, they pretend as the substrate and they bind to the enzyme. So there's no place for substrate to bind to the enzyme anymore. So as a result, the enzyme activity is reduced and the rate of reaction is reduced. But the key word for the competitive inhibitor is that it can be reversible. This reaction, the binding of competitive inhibitor to the enzyme can be reversible. And how can we do that? By increasing the substrate concentration. If we increase the substrate concentration, we can still reach the maximum rate of reaction. Let's look at the diagram. The diagrams are very important. So here, this kind of inhibition is reversible, and we overcome this by increasing the concentration of substrate. By the way, when you study the cell biology or even human biology, you don't study one sentence by sentence. You don't learn it by heart. What you learn is the concept. What happened first? And then what happened next? What is the result of this? What is the consequence? What can we do this? You remember them, you memorize them, and you write it in your own words. All right? You don't copy this sentence in your paper. You remember the concept only and you write it in your own way. This is easier for you to take the ASEN, and it will make you a lot easier for you to remember the cell biology. That is what I do. I don't really remember each and every sentence. What I do is like what happened first, what is this, what is it about. I try to remember first, I try to understand then, and then I try to memorize it. So that is how I do that. All right, right now I'm just telling you what are the most significant features and what are the most important concept that you need to write down if the question asks you about this competitive inhibition. So here, this line, green line is the rate of reaction. It will show the rate of reaction without the competitive inhibitor, and this one is with the competitive inhibitor. Look at the diagram carefully. Next, when we move to the non-competitive inhibitor, we will compare this graph and the graph for the non-competitive inhibitor. You can see that here, at last, both without the inhibitor and with the inhibitor reach the rate of reaction. Just now, he said that, comma, right? Name is comma. Okay, anyway, comma, okay. So, he just said that it only takes a little bit of time, yes, to reach the maximum rate of reaction is also true. Try to remember this graph, all right? In the ASEN, especially in MCQ, if Dr. Wong wants to test your understanding, she will give you a lot of graph and she will ask you which one is the graph with the inhibitor and which one is the graph without the inhibitor. You have to know that. And in order to know that, you need to understand the concept. And now let's move to the non-competitive inhibitor. So the working on non-competitive inhibitor is different. The shade of non-competitive inhibitor is not the same as the substrate. So it's not really competing with the substrate for the active site. What happened is that, right now we have another term called the allosteric site. What is the allosteric site? Allosteric site, the definition. Here, I think I'm showing you this one, right? It's the other part of the enzyme where the non-competitive inhibitor will bind. So that's called the allosteric site. So the key concept here is that the non-competitive inhibitor, it will not bind to the active site, it will bind to the allosteric site. So as soon as this non-competitive inhibitor bind to the allosteric site, the shape of the enzyme will change. The shape of the enzyme will change. So when the enzyme changes its shape, it will not be able to function anymore. Just now, the effect of temperature and the pH, I say that, in extreme conditions like high temperature, high temperature, high pH, in those conditions, the bonds in the enzymes are broken and the enzyme also changes its shapes. That is called denaturation. And the enzymes will not be able to function anymore. The same goes for this. When this non-competitive inhibitor bind to the allosteric site, what happens is that these enzymes will no longer have its normal shape and it will not be able to function anymore. So that is non-competitive inhibitor. And now let's look at the diagram, the graph. So can you see this? Without the inhibitor, the maximum rate of reaction is this much. But with the non-competitive inhibitor, the maximum rate of reaction is this much. So as soon as non-competitive inhibition bind to the enzyme, the rate of reaction will no longer be at its normal rate of reaction. It will reduce for sure. It will be reduced for sure. But let's look at again here. You can see that the maximum rate of reaction can still be achieved even with the competitive inhibitor. It's the same. It's the same here, right? But for the non-competitive inhibitor, it's not like that. The rate of reaction is reduced. Do you understand it so far? Any questions here?
No.
No, right? Okay. Try to practice drawing these graphs on your own and try to label it as well. And then what is more important is you need to understand the graph first. Because I think the other one likes to add those graphs in the MCQ. So if you have a basic understanding, you will be able to answer any types of questions. And also, these are the non-competitive inhibitors. It's good to know these as well. Even the Delta one will highlight this one, right? So try to know these examples. It's on page 47. I am telling you this so that when you do the revision just before the exam, you will know which page to go and which page to skip. You can possibly revise everything, every page of your lecture notes on your exam day. You can only go to the most important ones. And in my opinion, these are the most important ones. Right, so that is it. And then we have another one, allosteric inhibition. Just now we call this allosteric site, right? Please be careful. Allosteric site, allosteric inhibitors, and allosteric inhibition, and product inhibition. They are not the same. Don't confuse allosteric enzyme bind to the allosteric site. Don't confuse like that. So allosteric affinities mean let's say that we have the reactants A, and then we go to B, and then we go to C, and then we go to D. So as A is changing into D, the concentration of D is increased. So when we have a large amount of D here, this large amount will inhibit the A again. So that's the main concept. D is the end product, and this end product will inhibit the conversion of A into D. So that's what we call end product inhibition. And this enzyme, so this end product becomes the enzyme. That's called the allosteric enzyme. Is it clear? Try to read it on your own as well. Anyone clear, right? So this one, exactly. You don't need to know, you don't need to draw this one. It's just to make you understand. Okay, so that is it. That is the end of the enzyme. And let me tell you what you need to know for this. This whole file, you can have a lot of MCQ, you can have a lot of structure questions, and even the essay. So try to read through that carefully. And today, what you need to know is this one, the enzyme. Can you see the screen? The Google Doc. You need to know all the graphs here and the definition of AS, AE. AS is actually at this site. AE is activation energy, and you need to know the enzyme as well. And be careful, the enzyme doesn't increase the number of products. It only increases the rate of reaction. And two mechanisms of enzyme, what are they? They are induced fit and lock and key mechanism. And then another one is effect of pH, temperature, and the concentration on enzyme activity. Concentration, when it comes to concentration, we have substrate concentration and enzyme concentration. And then we have inhibitors and allosteric inhibitors. You need to know these 5 things. Right, so that is it. That is all for today. Do you have any questions?
No questions.
Great. If you don't have any questions, I will end this, and maybe we will meet again next week. And like I said, I would like to do chapter 2 in details. And I think next week I will do the genetic as well. Genetic is the most difficult one, right? Is it?
The genetic engineering?
No, genetic, not engineering. Or
Oh, genetic.
yeah, genetic recessive and dominant, right? There are
Oh, yeah.
some problems. I think they are quite difficult.
Yeah.
So we will go through that, and yeah, that is it. Wait,
Okay.
then have a good day. If you have any questions, just send me a message.
All right. Thank you.
Right, bye-bye.